Good afternoon, everyone. It's my distinct pleasure to introduce our esteemed speaker for today's seminar on complex lymphatic anomalies, Bill Polacheck. Bill completed his PhD in mechanical engineering at MIT in 2013, and following that, he completed a postdoctoral fellowship at Boston University and Harvard. In 2018, Bill moved to Chapel Hill to join the UNC and NC State Joint Department of Biomedical Engineering. And there he leads a dynamic research group focused on developing microphysiological and organ-on-chip models, which are vital for disease modeling and drug screening. And he's really, his research is really at the cutting edge of, I would say, biomedical engineering and making these in vitro models of lymphatic and vascular malformations. So his pioneering work is really notable in the realm of vascular and lymphatic malformations, where he's been employing his models to study cellular responses to mechanical forces such as flow. And Bill has received several prestigious awards, including the Young Innovator Award from the Biomedical Engineering Society, a Teaching and Mentoring Award from his department, and he was a recipient of a Miro Award from the NIH. And I also highly encourage everyone to take a look at his recent publications in Science Advances and APL Bioengineering, where he has explored in vitro models of PIK3CA-induced malformations. And I anticipate that he's going to elaborate more on this subject today during his talk. So the title of his presentation is Microfluidic Models of Vascular and Lymphatic Malformations. So please join me in welcoming Bill, and Bill, the platform is yours.
Great. Thank you, Mike. Thank you for that kind introduction. Yeah, so this presentation sort of weaves a little bit of cell biology and a little bit of devices, but the idea really is to introduce some of the tools that we've been developing. Our group is trying to improve on ease of dissemination of these tools. So if there's anything that's interesting to you, or if you think the models could benefit from further development, I'd be really interested in hearing from you. And also for those of you who are on now, feel free to interrupt me if you have a question about details that I maybe skip over, because I'm going to cover some material. As Mike said, I'm going to talk mostly about those two recent publications and sort of give a high level, and I'm happy to go into details as necessary. So I have a couple of disclosures to make. Most of them are not relevant except for the last one. We are working on some of the work at the very end. There's a provisional patent that describes, but I'll go into actually the details of that. We maybe submitted the provisional a little early, so I'm not actually sure that it's super relevant to the work in the end. But I thought I would introduce my group and our sort of philosophy a bit before going into the specifics. So I've been working in microfluidics for a long time. I guess I'm old enough now to say before they were really broadly called organs on chips. Mike Schuler was using that term, but nobody else really was when I started working in the field. And I started working in microfluidics from a purely fluid standpoint. The intention for my PhD was to really understand the physics of fluids on small length scales. I've always been really interested in fluids, but a guy next to me when I was an early graduate student was running a rheometry experiment where there were living cells, and the curves and stuff that he was getting out of the data were so interesting that I talked to my advisor about maybe we should modify our microfluidics to look at cells, and all of a sudden one thing led to another, and I've been working on cells ever since. But the advantage of microfluidics from thinking about cell biology now with this big push for novel and alternative methods from NIH and FDA is that they afford high precision of control of fluid pressure and flow. And they also allow us to modulate solid stress and strain. So we have pretty good control over the mechanical microenvironment of cells in these systems. They also allow us to pattern spatially at resolutions that are much smaller than a single cell. So we can assemble adhesive and non-adhesive areas, and we can start to look at how cues from the microenvironment affect cells in a sort of spatial way. In theory, they're high throughput. most of the ways that we actually make these in the lab are not that high throughput, but that's a big effort of our lab right now is to try and actually push these out at rates where we can start to do real drug testing, particularly in a patient-specific manner. But a challenge with these devices is that if you assemble them in sort of a traditional microfluidic approach, cells are often in contact with very stiff surfaces. That's why I include these images here. Either silicon on the left, glass on the bottom here, or hard or stiff elastomers like PDMS, which form the top of this microfluidic device. On conversely, three-dimensional biomaterials sort of address some of these problems. By allowing the sort of recapitulation of the native biochemical and biophysical microenvironment in vitro. These are materials that are made from either animal components or human components or synthetic polymers that are engineered to behave a little bit more like native matrix. One big advantage, particularly in studying vascular malformations, is that these materials allow cellular remodeling. If we design them right, the cells can invade and they can remodel the matrix, much as a lesion does during progression, provide some level of tissue architecture as well. So our lab's approach has been to try and merge these two, to try and make microfluidic devices where we have 3D biomaterials inside. There's a lot of groups doing this now. It's a little harder than it sounds, as a lot of engineering is, but one of the biggest challenges is it takes a lot of pressure to push fluid through tiny channels. And if we want compliant biomaterials that have mechanical properties similar to what's seen in vivo, they tend to stretch a lot under those pressures. So there's a lot of stuff that goes on in the back end in terms of optimizing pressures and assemblies of these systems that I'm happy to address. We try to be really forthcoming with these challenges, but still some of the details get swept under the rug in publications. Just a brief overview of like one example of how we make these devices. In my lab, we have probably 20 working different models, but this is just one kind of standard model. But I just want to introduce it broadly in case you're interested in adapting it. But the way that we make these microfluidic devices is we borrow techniques from the semiconductor industry, like really, really easy versions of what they're doing these days. But basically, we pattern silicon by using a series of photosensitive polymers that we can put onto silicon wafers with very precise thicknesses. And then we put on transparency masks on top of that and we shine UV light. So anywhere where there's a hole in the transparency mask, light gets through to the photosensitive polymer and induces cross-linking to make it insoluble in some solvent. And if we can layer these patterns on top of each other, much in the way that you would for 3D printing. And then at the end of this process, we can wash away all the uncross-linked stuff, and you end up with kind of an extruded version of your transparency masks. But we use these silicon wafers. We don't use their semiconductor properties. We just use them really as a mold to then pour on polymers that we can cross-link, something like PDMS. We use other materials depending on our applications as well. We can peel it off, and anywhere that was raised on the silicon wafer becomes a channel in the resulting PDMS device. And we can bond these to glass and then enclose them. And so we end up with these geometries where the characteristic length scales depend on the transparency mask, and we can get those down to on the order of less than a micron, if necessary. We don't ever go down that low. But where the sort of unique approaches that our group takes are, this is really just the starting point. Then we inject hydrogels into the device. We can do so just as a bulk hydrogel. We can preload the hydrogel with cells. There's several different configurations, but in this specific case, and we use this model later, which is why I introduce it here, we inject a hydrogel around something like a needle or a sacrificial material that we can then pull out or dissolve away, and we end up with a hollow tube through the hydrogel. We can then backfill that with cells, in this case endothelial cells, and then we end up with tubes that we can then hook up to various flow systems to control pressure and flow through the middle. Again, this is kind of the simplest model. We can layer on different cells. We're working now on alternative fabrication techniques to try and make things that look a little bit more like arteries and have the kind of characteristic wall structures of the artery. So this is just a cool space age video that was made in collaboration with Wes Legant here at UNC using light sheet microscopy. You can see we get these nice tubes where there's a continuous layer of cells around. So I'm going to use this as a model for blood and lymphatic vasculature in this talk, but we've also used this for epithelial tissues such as the bile duct and components of the liver as well. One of the key assays that we can run with this device is we can look at not just how the cells behave on different levels of perfusion, but also we can measure the permeability to different solutes. So here we're looking at top down at a vessel. So this is a tube and we're looking at the top down, which is why it kind of looks like a rectangle. And in the top case, we've applied flow for 24 hours. You can see the cells align quite nicely. VE cadherin, this is a molecule I'm going to come back to several times throughout the presentation as a marker for adherens junctions or cell-cell junctions in these endothelial cells. And you can see each cell, each nuclei is surrounded with a nice continuous layer of VE cadherin. But if we interrupt flow, or if we keep them static to begin with, very rapidly these cells sort of, the cells start to rip apart from each other and we get big holes in the matrix. And this is sort of this starting point, this observation that if we precondition them with flow, we get these really nice tight vessels. But if we stop flow, they sort of open up and degrade. That is actually the basis for when we started to think about mechanotransduction in vascular malformations, that I'll get to in a second. But one of the ways we can quantify sort of the cohesiveness of these vessels is we can introduce fluorescent markers. We can also take electronic readings. That's something we've been doing lately, is using impedance rather than these fluorescent tracers because it's a little bit higher throughput. But here's a representative image where we have 70 kilodalton dextran. It's about the same size as blood serum albumin, but it's fluorescent. We introduce it through the lumen of the vessel. And in the cases where we have flow, you can see it stays mostly inside the vessel. The warmer colors are higher concentrations. But in the case where we, I don't know why it's not playing well. There we go. In the cases where we then stop flow for some period of time, in this case 15 minutes, you can see that the vessel becomes leaky and this dye comes outward. And we can just quantify the rate of extravasation of this dye and formulate a diffusive permeability as a parameter that we like because we can compare it to in vivo. And that's sort of a baseline assay that we'll use moving forward. Another benefit of this technique is I showed just one single vessel, but we can start to array several vessels very easily. It requires us to change the physical microfluidic device, but that's actually not that hard. And what we can do in this paper was mostly a proof of concept paper, but we can pattern blood and lymphatic vessels in parallel. Here, they're kind of far apart, so you see them imaged independently. The blood is on the top and lymphatic is on the bottom down here. But from a sort of pharmacology standpoint or a tissue physiology standpoint, we now have sort of the basic transport unit of a solid tissue where we can introduce stuff through the blood, let it transverse this kind of interstitial tissue we could load up with cells and be drained by the lymphatics. And this allows us to look at both chemical transport, but also physical forces involved with flow through blood vessels or through the interstitium and drained by the lymphatics. So we had been working on these models when Dr. Julie Blatt, who was a clinician, she recently retired, here at UNC, approached us. Julie is very active in the vascular anomalies world, and for any of you who've met her, she's awesome, but she's very enthusiastic. So it was very close, very soon after starting my position here at UNC. I don't know how she found me, but she came and found me and she said, you need to be working on vascular malformations. And the reason she approached us actually was based on some of our work on Notch signaling. So my group's very interested in the Notch family receptors and how they are responsible for mechanotransduction of fluid stresses. And she was interested in repurposing some Notch inhibitors potentially for treating these patients. And we started to work on a grant together. And as we were working out the details of the grant, she realized actually what they need is not access to the drugs. We could get our hands on Notch inhibitors pretty easily, but what we didn't have was a testing platform, an in vitro platform where we could quickly screen concentrations and different drugs themselves to see if they have an effect on these cells that harbor mutations that may induce vascular malformations in patients. So through those conversations with her and a really awesome research assistant professor in my lab, Wen, who recently has moved on to the USPTO. But she, at the time, was interested in rare diseases. And so she took this idea that Julie suggested and really ran with it. And where this started was we took some of these known mutations in PIK3CA that are known to be associated with vascular and lymphatic malformations, and we introduced them into healthy endothelial cells. In this case, we used HUVECs, but now we're moving on to actual patient cells, and I'll get to that at the end of the presentation. And then after introducing these constructs that express the mutant forms of PIK3CA into the healthy cells, she introduced these cells into microfluidic devices to try and grow what kind of look like capillary networks or vascular networks. And initially, our goal was just to see, do we have a quantifiable phenotype that we could use then to screen, in this case, originally for Notch inhibitors, to see if they're effective at reverting vascular malformation phenotypes. So I'm going to skip over some of the details here and just show some of the nice images that Wen collected. The paper here was published in 2023, and that describes a lot more biochemistry than I'm going to go into today. But what I wanted to highlight was these cells here. There's two panels on the left, what's labeled as vector control and this PIK3CA with the E542K mutation. These are what the cells look like when they're in 2D culture. So this is just on tissue culture plastic. And you can see there's some differences between the cells for sure. Like these cells are a bit more crowded. They may be growing on top of each other. In some cases, they have very irregular cell shapes. And that had been well described in the literature. But relating these cellular phenotypes back to the actual malformation lesions is difficult. However, when we introduce these cells, initially we were using a fibrin hydrogel. If we introduce these cells into fibrin, you can see these panels are the endothelial cells. You can see they form very, very different structures. So the GFP, the infection control case, form these kind of tubes. This is early on. We can sort of let this go a couple of days. These become perfusible, and they look a little bit like a capillary network. Whereas the mutant cells form these really blobby, sort of open structures, and they degrade the matrix like crazy. You can see that a bit better in these 3D reconstructions where we just kind of take surfaces of the labeled cells. And you can see, this is a control line where we just overexpress the wild type form of PIK3CA under the same promoter as these two, which are mutant forms. You can see the wild type, we kind of have these. Maybe they're tube-like structures. They certainly look like a network of some sort, whereas the other two cases are very blobby and also very thin compared to this wild-type case. And that thinness is because these cells that express the mutation are actually eating through the matrix really, really fast. So I actually think Wen was presenting this work maybe in this exact forum. I think it was this exact forum a few years ago. And we were kind of worried about this because we were like, man, these cells degrade the matrix so fast that it's very hard for us to actually capture the cellular phenotypes because they just get down through to the glass very quickly. And somebody in this forum was actually like, that's actually a very helpful phenotype to quantify because we know in vivo that these cells tend to be highly proteolytic, or at least areas around the lesions, there's a lot of proteolysis. So perhaps this is something that we could dig into. And that indeed is a direction that Wen went in this paper, and she looked at a bunch of inhibitors and signaling pathways to try and relate the sort of blobby cellular phenotype to matrix degradation. And I'll refer you to that paper for those details. But what I wanted to talk about today and focus on is some of the work that we've been doing to try and understand the role of the microenvironment in lesion progression. As Mike mentioned, my background is in mechanical engineering. I'm really interested in forces and how cells respond to mechanical forces. And it's quite interesting that in the certain classes of these vascular and lymphatic malformations, looking through the literature and talking to Julie Blatt and Alex Borst, another clinical collaborator here at UNC, they describe the lesions as soft and malleable. So there's there's a biophysical phenotype to them, and also that these specific PIK3CA associated lesions occur in low flow environments. And these are environments in which perfusion rates are low. And so there's a kind of chicken and the egg question here is the low flow environment due to this overconnected vascular network that's characteristic of the lesions, or is there something about the low flow environment that sort of potentiates progression of these lesions itself? It's probably coupled to some degree, but our real question was is does the low flow environment permit growth of these lesions in a way that might provide some cues as to maybe how to slow the progression in the long run. So we were interested in sort of two specific questions, and that is, do endothelial cells that express these PIK3CA mutations respond to hemodynamic shear stress in particular in a manner similar to wild type cells? And if there is a difference, could the dysfunctional mechanotransduction play a role in lesion progression? So we did a series of experiments to ask this question. And to be honest, the results aren't always, it's not the cleanest picture that we have. But in trying to ask these questions, we've developed a number of tools that I'd like to highlight in case they're interesting in your own work moving forward. So the first experiment that we did just to probe mechanotransduction of these cells was, I hate this experiment as a mechanical engineer, but it's highish throughput. So my lab has pushed back on me for years, and I finally have come to accept this as a very helpful first tool in looking at mechanotransduction. And it's pretty straightforward. We just cultured these cells. We cultured both the wild type expressing cells and the mutant cells in multi-well plates, and then we put them on an orbital shaker. And when you put them on an orbital shaker, if you get the height of the fluid right and the rate of sort of orbital shaking right, you can get these flow profiles where towards the edge of the plate or edge of the well, the flow looks pretty laminar and the cells will align, which is kind of the gold standard of functional mechanotransduction of endothelial cells. But at the center, we have something maybe like disturbed flow. It's kind of hard to predict. It's certainly not laminar and unidirectional. And so what's helpful about this is in a single well, you can sort of scan from the center to the outside and look at how increasing laminar flow might affect the cells. And since it's a multi-well plate, we can test multiple cell types in the same dish and have nice control. And we can do that in a way that's a lot faster than any of the pump-driven flow systems, which we always use to check our results here. So you see on the top case what happens in these PIK3CA wild type cells. There's actually a lot of information packed in here. The cells are stained at the end of the experiment with V-cadherin antibody that's shown in magenta. DAPI shows the nuclei. But then yellow is this streptavidin assay that we do to assay cellular permeability in two-dimensional configurations like this one. We first coat the plate with biotin. It's like fibronectin functionalized with biotin. And then before we fix the cells, we add streptavidin for just 5 minutes or so. That's fluorescently tagged. And then we wash everything off. And so any streptavidin that gets through the endothelial monolayer then binds to the biotin underneath. It stays there. And that shows us basically holes in the monolayer. So what you can see is that at the center where these cells are getting the kind of more disturbed, less laminar flow, we see these big yellow regions. That means there's breaks in the cells, there's leaks there. But as we start to progress out towards the edge, there's fewer and fewer of them, and they're localized to these tricellular junctions, which tend to be the leakier components of a monolayer. But if we do the same thing with the mutant cells, you can see there isn't a decrease in the area of these yellow spots. So here, it's not so much the intensity we're interested in, it's the area. We normalize these in a way from a single experiment so that the intensity of the yellow signal might vary from condition to condition, like here to here, but the area gives us an indication of how leaky the monolayer is. And if we quantify that, what we see is that in the wild type cells, they both elongate. We measure that through eccentricity, meaning like how long a cell is relative to how wide it is. The wild type cells elongate as we go out towards the edge where the flow is laminar, and they get less leaky as you go out towards the edge. Whereas those cells expressing the mutation, they don't elongate and they also don't get less leaky. So there's a problem with mechanotransduction here, or there's a problem with the sensing of flow. We're not sure. But we focus on VE cadherin. Actually, if you look closely here, for example, you can see that there's actually a break in the VE cadherin at cell-cell junctions. And one of the reasons we're interested in that is VE cadherin plays an interesting kind of dual role in mechanotransduction. It itself is a flow sensor. It responds to flow. It's required, its expression is required for endothelial cells to respond appropriately to flow and to align response to flow. But also it mediates cell-cell junctions to establish barrier function and importantly to coordinate forces across the monolayer. So it helps the vessel behave like a vessel rather than a collection of individual cells. This experiment doesn't allow us to decouple those two things. So Wen did a cool experiment where she infected these cells with inducible mutants, either an inducible wild type form of PIK3CA or an inducible mutant form. And this allows us to turn on the mutant before, after, or during flow. So there's a lot of images here, but you see the column is the center where the flow is not laminar. The right column is the edge where the flow should be more laminar. And then we go from untreated, so these should not have expression of the mutant, to adding doxycycline before flow, which should turn on the mutation before flow, with flow, or after flow. It's a little easier if we start to quantify, but one of the interesting observations we had is that the cells, if we turn on doxycycline to induce the mutation after flow, they maintain their alignment to flow. So in other words, they've sensed flow, they've started to align, we turn on the mutation, they remain aligned, but they don't maintain their barrier function. So if you look at these curves, this is a quantification of percent of streptavidin area across the well plate. You can see that before and after flow, there's not much difference, meaning they're just as leaky whether that mutation is expressed before flow or after flow. And they are leakier than the case where we don't induce the mutation. So there's some separation here between the cell-mediated alignment and the barrier function of the endothelial cells. And this caused us to further think about the role of VE cadherin in these responses to flow. Now, one of the interesting components of mechanotransduction at VE cadherin or adherens junctions more broadly is that this similar process happens in focal adhesions, but there is a series of protein-protein interactions that occur at the application of force across these VE cadherin containing cell-cell junctions. And one of the early stages is that vinculin binds to alpha-catenin at sort of the cell side of VE cadherin, and that stimulates the aggregation of more proteins that results in a plaque that ultimately connects to the actin cytoskeleton. So we were interested to see if perhaps vinculin binding was disrupted in these cells. In other words, we know that there is a barrier function defect that we think is via VE cadherin in response to flow. Is there a problem with vinculin getting there? That would kind of help us figure out where the dysfunctional mechanotransduction is coming from, whether it's the cell sensing of flow or whether it's the establishment of barrier function. It's a little bit hard to see if you're not used to looking at these images. But if you look at these, the HUVECs, these are GFP transduced as an infection control, you can see a thin line of vinculin at the cell periphery. This is vinculin that is co-localized with VE cadherin. Now this is the wild type case, but these are co-stained, and you can see that there is sort of a trace of vinculin along VE cadherin containing junctions. But in the case of the mutant cells, we lose the association between vinculin and VE cadherin, and actually we still get it at these puncta, which are the ends of stress fibers. So this is telling us that perhaps these junctions, maybe there is VE cadherin at the periphery of these mutant cells, but they're not being solidified. They're not the intracellular plaque proteins that stabilize VE cadherin are not assembling appropriately. And concurrent with that, we also observed, so we looked at a protein expression of these various plaque proteins, and we saw that vinculin in particular and beta-catenin, these are not differentially expressed in the two mutants. So this is a protein aggregation issue, not a protein expression issue. And interestingly, we also saw a downregulation of phosphomycin light chain 2, which is one of the molecules that regulates cell-generated forces. This is perhaps pointing to dysfunctional intercellular force communication between these cells. And one of the other things that force across VE cadherin containing junctions does is it stabilizes VE cadherin. It prevents its internalization. So to see if maybe this destabilization or the lack of vinculin binding did lead to destabilization, when did a pulse chase experiment where she just added a pre-conjugated fluorescently tagged V cadherin antibody for a short period of time to mutant cells versus wild type cells. It's a little hard to see here, but there were a lot of these puncta in the mutant cells, which is indicative of, actually the live images are better. I don't think I have them here, but anyway, it's indicative of increased recycling of VE cadherin at the cell membrane. So this suggests perhaps that the junctions are there, but they're remodeled much faster. And perhaps that's due to a lack of vinculin binding. Okay, but all of this points to perhaps that these cells are not bearing force across cell-cell containing junctions. And that led us back to this model that I introduced at the very beginning of the talk, because there's a interesting aspect of the vessels and what's required to establish barrier function in these engineered models that I'll explain in a second. But what Wen first did was take this vessel model that I introduced earlier, and she put in these different cell types. And what's really interesting in a different vessel is what was sort of on the cover of the talk, the bright yellow image on the title slide, is that when we introduce these mutant cells, and this isn't just true for the E542K, we've done a couple of other mutants as well, and they all sort of look similar. They like explode out of this vessel. This is just 24 hours after seeding. And remember here in this configuration, black is actually a three-dimensional collagen hydrogel, so the cells can invade. It's not empty. It kind of looks empty here. But you can see that these mutant cells start to invade like crazy. They increase the lumen size. There's some migration away from the vessel itself. And if we treat with rapamycin or alpelisib, we revert some of those phenotypes. Maybe alpelisib is slightly more effective at the doses that we tried here, but it blocks some of the invasion. There's a lot of actin debris in the matrix. We're not exactly sure what's up with that, but in general, treatment with these two drugs sort of reverts these phenotypes, which is further suggestive that it's the mutant itself that's driving this. But getting back to this force coordination question, one cool thing that we've noticed over the years in these vessels is that if we start with patterning, let's say 160 micron tube through the collagen gel, that's our sort of standard starting point. If we pattern 160 micron tube and then we seed in endothelial cells, if they're healthy and they form a good barrier, they shrink the tube down a bit, not a lot, but maybe from 160 to like 140 microns. And that shrinking, that cell-generated force that's associated with the shrinking is necessary for barrier function. If we perturb the system in any way that blocks that shrinking, we don't get good barrier function. So to sort of further explore our hypothesis that something's going on with coordinating cell-cell or forces through cell-cell junctions, Wen did a kind of clever experiment where she seeded these devices with wild-type cells or with these mutant cells, and then she imaged the matrix using confocal reflectance microscopy before and after decellularization. This is kind of similar to what's done in traction force microscopy, but rather than tracking individual beads, we can just track changes in diameter of the vessel. And what we found is in the wild type cell, there is some relaxation that happens when we decellularize. That's the purple after phase. That shift from the cyan to the purple here is indicative of how much stress the endothelial cells were applying before we remove them from the system. It doesn't look like much in this image, but if we quantify it, we can see that there's a pretty significant difference in displacement between the wild type case and the mutant case, which does not cause much relaxation at all. So this is another point towards the fact that perhaps there is an issue with coordinating forces among these cells in the monolayer when they express these mutations. And I just wanted to bring back to this point that if you look at this specific example that I had shown earlier from a previous publication of ours, you can see that when there's flow present, we see this sort of shrinking of the vessel. The diameter of the flow condition is less than the diameter of flow plus static, and also that there's a lower permeability in the flow condition. So what we were kind of interested in is if the mutant cells aren't contracting the vessel, maybe if we stop flow, they won't rip apart from each other either. So in the mechanobiology world, there's this concept of tensional homeostasis, meaning cells are in mechanical equilibrium with their environment. If they aren't sort of contracting in response to flow, perhaps they also won't relax as much if we remove flow. Maybe we won't get the holes and stuff here. So we did an experiment where we took the wild type expressing cells first. We introduced flow for 24 hours. We let them become sort of accustomed to flow, and then we stopped flow for just an hour. And actually, we've done this over several different static durations, but in this case, it was just an hour. And you can see even within an hour, these big holes start to open up in the wild type case. And this is sort of consistent with the previous slide. But if we do this with the PIK3CA mutants, yeah, it's not a tube anymore. It's this blobby mess. But when we stop flow, we don't actually get any of those holes. And if we quantify the gap area, in this case, that's the most interesting one. The cell number doesn't really change in either of these cases, indicating the cells are kind of ripping apart, maybe not dying, though, in this case. But if we quantify gap area, we see a big increase in the gaps in the wild type case, and not so much of an increase or no increase at all in the wild type or in the mutant cells. And this suggests these cells are somehow preconditioned to this interrupted flow or the low flow environment that might be characteristic of the lesions in vivo, and gets back to our overarching question that we sought to address here. And one other interesting aspect of this kind of flow-induced homeostasis in wild-type vessels is that it's known that flow counteracts angiogenesis. So in other words, the induction of homeostasis that's associated with hemodynamic shear stress sort of makes it harder for cells to undergo angiogenesis. It's a quiescence signal. So another question we had is perhaps if these vessels are not flow conditioned, like the mutant vessels are not flow conditioned the same way wild types are, maybe they're more permissive to angiogenesis. And as mechanical engineers, we're also very interested in how the pressure associated with these low flow environments might regulate angiogenesis as well. So we kind of did this double experiment next, where what we did was elevate the pressure both internal to the vessel relative to outside, and then vice versa. So in this case, the vessel sort of acts as a source, and fluid flows from inside the vessel out into the surrounding matrix. That would be like a pressurized vessel. In the second case, what we did was introduce flow from outside to in. This has been shown in many different instances to induce angiogenesis. It's something that Judah Folkman actually was interested in, and its role in angiogenesis as well. And you can see that when there is this flip, this basal to apical flow, as we call it, this draining flow, there's a little bit of sprouting in the wild type case. But in the mutant case, it goes crazy. These cells invade all over the place on both sides of the vessel, but much more significantly towards the source of pressure. So this is again consistent with this concept that maybe the cells aren't getting these flow-induced sort of homeostatic quiescence signals. They're not transducing the flow correctly, and it's leaving them in a state where they're much more amenable to angiogenesis or sprouting growth in these kind of low flow environments. So we also went into, I want to be conscious of time and leave time for questions. We went into some of the mechanisms as to why these cells might invade more. And this kind of, the biochemistry eluded us a bit. So we did some physical characterization. And interestingly, I don't have it on a slide, but I have it in a supplement if anybody's interested. We find that these cells are much more compliant. So we did some micropipette aspiration. We found that cells expressing these mutants, both the cell itself and the nucleus, are much more compliant, which should make it easier for them to migrate through the pores of the extracellular matrix. We don't know why that is yet, but we're thinking that perhaps that's a, either consequence or cause of the increased migration that we see here. The other thing that I did want to just leave one slide on to add a little bit of translational context to some of this work is that another thing that flow does in a healthy vessel is it suppresses endocytosis. So typically, you have this upregulation of endocytosis very quickly after you initiate flow, and then it suppresses in a healthy endothelium. So one of our hypotheses was perhaps if these cells aren't responding to flow correctly, there is upregulated endocytosis at baseline for in these mutant cells. And if there is, perhaps that's an opportunity to deliver some drugs via something like a nanoparticle or something. So we did a series of experiments. I'm just showing one slide here, but we've done a lot more now with inhibitors and characterizations where we add both polymeric but also lipid nanoparticles in collaboration with Owen Fenton here at UNC. And I don't. This is an overclaimed targeted therapy. There's no therapy here. These are just nanoparticles. And we tried different sizes, but around these 110 nanometer particles, what we see is that even in the conditions of flow, we see a downregulation in the uptake of these particles in the healthy cells, but there's still larger degree of uptake in these mutant cells. These are just confocal images, but we've now done this with flow cytometry to show that these cells are actually internalizing the nanoparticles. And so we're hopeful that this is just based on size. We're hopeful that with a little more engineering, we might be able to take advantage of this to get drugs more selectively inside the mutant cells than in maybe surrounding healthy bystander cells. So that's time. I'm a little bit over. I apologize for that, but just as a summary, some of these tools do seem to give us some phenotypes at least that we can screen for both drugs, but also potentially mechanism. One thing that we're doing now with Alex Borst, who recently moved from Penn down here to UNC, is she is helping us get our hands on some patient cells so we can get away from these, studying mutations that are already pretty well known and into actually dealing with the difficulties and the heterogeneity of patients. And we find that there is some kind of dysfunctional mechanotransduction going on here. These cells don't really respond to flow as healthy cells do. And one kind of project that I'm pretty excited about is there's a collaborator here at UNC who does kinome screening. So we're looking now at what different kinases, the different kinase activities with and without flow in healthy and mutant cells to see if there's anything potentially targetable. And as I mentioned, sort of at the very end, we're also thinking about how this dysfunctional mechanotransduction might allow us to take advantage of endocytosis for targeting these cells. So I just want to thank our collaborators. I want to thank the whole broad vascular malformations community. The community has been fantastic, and it's really funding from LMI and the $1 Million Bike Ride that allowed us to do any of this stuff. We wouldn't be in here, and now our group is really excited about this, and we're hoping to continue to develop these models and work more towards translation as we move forward. And with that, I thank my group. That's some combination. I can never get everybody in the same room, so it's some combination of these pictures is the current group. But I'm also happy to take any questions or clarify anything. If you have them now, you can also email me directly if you're interested in any of these models, or if you have big problems that you want to tell me about, I'm happy to hear them too.
That was a wonderful presentation, really exciting work.